Video 13.1 kicks off our unit on biotechnology. Biotechnology is when biological systems are used to accomplish some other task to benefit people. In other words, once scientists understand how biology works, they can manipulate biology to do things like make insulin, diagnose and treat diseases, determine evolutionary relationships, and even solve crimes. Video 13.1 will focus on viruses. First, we'll talk about the biology of viruses so that we can lay the groundwork for how viruses can be used in biotechnology. In this video and in our class, we will focus on this chunk of objectives. Pause for a closer look. Some examples of viral infections with very visible phenotypes are shown here. Now, in addition to these, we've heard about HIV, the flu virus, rabies, and coronavirus, to name a few others. When you look at virus particles, they seem very diverse, but the fact is that all viruses share a simple template of structure and function. And once you understand this simple template, you'll be able to understand all the different ways that viruses work. Regardless of the viruses, all viruses share the structure of having genetic material, DNA or RNA, surrounded by a protein coat called a capsid. In each of these four viruses, RNA is shown as red, so we've got 2 RNA viruses here, or DNA as genetic material is shown in dark blue, so we have 2 DNA viruses here. Then, every virus, regardless of whether it has DNA or RNA, has a protein capsid shown in light purple. And some viruses, like the flu virus shown here, are surrounded by a phospholipid membrane called a viral envelope. One way that viruses can be classified is by the type of cell the virus infects. Some viruses specifically infect prokaryotes, while others infect eukaryotic cells. Let's start with an analysis of prokaryotic viruses and build from there. Prokaryotic viruses are called bacteriophage, or simply phage. Phage look particularly sinister because of the most famous phage has tail fibers that look like legs and make this whole thing look like a creepy crawly. Still, the structure follows the basic template, genetic material inside of a protein capsid. As shown here is a bacteria getting infected by over a dozen phage. Now, phage get their name because they eat through bacteria. The left side of this plate of this petri dish is completely covered by a cloudy lawn of bacteria. After infection with phage, the phage kills the bacteria and appears to just eat all of these holes through the bacteria on the plate. Now, phage have 2 different strategies when it comes to infecting bacteria. The first strategy is called lytic infection, which is shown here. Now, to better understand this process, you can break it up into 4 steps: infection, synthesis, assembly, and release. Let's look at each step in more detail. First is infection, how the virus gets its genetic material into the host cell, shown in these steps up here. Now, viruses identify host cells with a lock and key mechanism, where viral surface proteins fit like a key into the lock of specific receptor proteins on the outside of host cells. This lock and key interaction determines which species and which cells a virus can infect. Most viruses can only infect one or a few species, and many viruses only infect certain cell types in certain species. All right, so after attachment here, the viral DNA enters the host cell. Viral DNA is shown in blue, and that completes the infection process. So in the case of a phage, this protein coat, the capsid, stays outside, but a lot of viruses work by having the whole virus go inside of the host cell. The next step is viral synthesis, where the virus copies itself inside of the host cell. Now, the virus genetic material is duplicated. You see a whole bunch of copies in blue here. That genetic material is transcribed into RNA and translated into proteins. So that whole central dogma thing of protein synthesis happens. But in all of these processes, the host provides the nucleotides, enzymes, ribosomes, tRNAs, amino acids, ATP, and all of the other components needed for these processes. So essentially, the virus hijacks the host cell, turning the host cell into a virus factory. After synthesis is assembly, where newly formed virus pieces are put together into virus particles, shown in more detail here. You could see how the phage DNA is packaged into the head proteins, and then all of the other proteins are attached to make the complete phage during the assembly process. Now, finally is release, where the newly formed viruses leave the host cell, and then they can go on and infect other cells. By the end of the lytic cycle, a single host cell has been taken over to produce hundreds of virus particles. Now, the lytic cycle gets its name from the dramatic cell lysis, bursting that happens at the end of this process, exploding the host cell to release all of these hundreds of new viruses to infect more cells and keep this cycle going. The other viral life cycle is a little more low-key, but perhaps more sinister, and it's called the lysogenic cycle. Now, lysogenic infection is a little more sneaky, and the virus actually integrates its genetic material into the host. Let me explain. So if you look at the part on the left here, you can see the familiar lytic cycle like we just talked about. On the right, though, you can see what's different about phages that use the lysogenic cycle. So the phage infects the host bacteria on the top and releases its viral DNA into the host. Next, with the lysogenic cycle, you can see that viral proteins open up the host DNA and integrate the viral DNA into the host DNA and then religate everything together. Now, the term for phage DNA that's incorporated into the host DNA is prophage DNA. So what's new about this is integration. After integration, you can see that when the bacteria reproduces by fission, the prophage DNA is copied by DNA polymerase along with the host DNA. And the end result is that every daughter cell has viral DNA incorporated into its own bacterial DNA. How sneaky is that? So the phage DNA remains silent in the bacteria DNA. Now, under the right environmental cues, typically some sort of stress, the prophage DNA can release from the bacterial DNA and then trigger a lytic cycle. Then the host bacteria is turned into a phage factory that eventually releases hundreds of infectious viral particles, just like we saw before. So the lysogenic cycle is kind of like the lytic cycle with this bonus sneaky phase attached. Those infection cycles make phage seem completely unstoppable, right? But the fact is that bacteria have a few defenses of their own. The two that I want to focus on are restriction enzymes and the CRISPR-Cas system. Down here, you can see that restriction enzyme, shown in green, attached to phage DNA, which is shown in red. Restriction enzymes are nucleases, so they hydrolyze the phage DNA, cutting it into bits. Restriction enzymes recognize specific DNA sequences only found in phage, so they won't damage their own bacterial DNA. Now, the CRISPR-Cas defense mechanism works in a somewhat overlapping way. In short, a small piece of phage DNA is saved by the bacteria in its own DNA. So now if the bacteria survives the phage infection, this piece of DNA acts as kind of like an immunological memory. So if the phage comes back, the CRISPR-Cas system specifically hydrolyzes phage DNA, which is shown up here. Now, we'll get into more detail about these later, but for now, know that both of these defenses act like scissors that can specifically cut the viral DNA. All right, so that's it about prokaryotic viruses. So they're the most studied and have given the best insights about how viruses work. And eukaryotic viruses are a little bit more complicated, but are still just variations on the simple themes that we saw with phage. There are many different ways to classify eukaryotic viruses, but they're still just a protein coat with genetic material inside. Some eukaryotic viruses have a viral envelope, which is a phospholipid membrane that surrounds the capsid. And eukaryotic viruses can also have DNA or RNA genetic material, and that can either be single-stranded or double-stranded genetic material. So what's the big deal about having a viral envelope? A viral envelope is an outer phospholipid membrane. There are viral proteins embedded in the viral envelope that are shown in purple here, and those viral proteins bind the virus to the host cell, just like the lock and key mechanism we saw in phage. What happens, though, is that the viral envelope fuses with the host phospholipid membrane and releases the whole protein capsid and viral genome into the host cell. Now, just like with phage, the virus reprograms the cell to make viral particles, but here's the interesting part. Viral envelope surface proteins that are being made over here in the endoplasmic reticulum, since they're glycoproteins, those viral envelope surface proteins are put onto the host cell membrane using transport vesicles and exocytosis, and then when the assembled capsid and genome leave the host cell, they actually bleb off a piece of the host cell membrane on the way out. So, in other words, viral envelopes are derived from the host membrane. And these viruses then would have host cell phospholipids and host cell membrane proteins too that may help the viruses avoid detection by the host immune system. The other thing that's different about the virus in this picture is that this virus has an RNA genome instead of the DNA genome that we saw before. And you can see here that the virus genomic RNA is a template to make RNA, mRNA, to make new viral proteins, but the RNA genome is copied and packaged into the capsid of each new viral particle as well. So why use RNA instead of DNA? Now remember, natural selection favors organisms with immune systems to stop viruses. So viruses have to keep evolving too. It's like a constant back and forth. And the thing with RNA is that the host's RNA polymerase that's used to make all of these copies doesn't proofread as it makes copies of the viral genome. So as a result, the mutation rate is much higher in viruses with an RNA genome compared to DNA. So after infecting a host, the daughter virus particles will have much more genetic diversity and increase the chance of virus evolution. Maybe a new mutation helps the virus evade immune detection or increases the ability of a virus to find a new host. That's pretty crazy. All right, here's a recap. Despite their diversity in host cells and genetic material, all viruses have the basic structure of genetic material surrounded by a protein capsid. Viruses have a couple basic strategies of infection. First, lytic infection, where a host cell is immediately taken over, and a lysogenic cycle, where viruses insert their genome into the host and wait for just the right time to take over. Bacteria have evolved the CRISPR-Cas system and restriction enzymes to cut up phage DNA and prevent infection. Eukaryotic viruses can have the additional features of a viral envelope and RNA genome. Viral envelopes add a layer of protection to the capsid and genetic material and may help the virus avoid immune detection. Now, a big advantage of an RNA genome is that RNA mutates more rapidly and can result in faster virus evolution.